So you're going to have to know the difference between meiosis and mitosis. This is something we've gone over before. But they both result in new cells being formed. So that's good. And those phases that we learned before, anaphase, telophase, prophase, metaphase, those phases, that verbiage is still used for both processes. Now microtubules are used. You have chromosomes doubling before separation. So I'll probably strike that about the chromosomes. So chromosomes, I'll say 2x. So they double before it happens. Now the difference is meiosis, you have crossing over that happens before, and that creates new combinations of genes. And the new cells you make, this is critical. So listen, the new cells you make from meiosis only have half the number of chromosomes. So for example, in humans, we have 46 chromosomes. When we go through meiosis, our resulting cells have 23. So it halves it. And mitosis, it's 40, say 46, it goes to 46. So mitosis is strictly for copying cells. One cell makes two cells, and that can be, in meiosis, that can be different. Sometimes one cell makes one cell, or one cell makes four. Now this would be in oogenesis, one cell creates one egg. Spermatogenesis, we have one cell making four, one pre-sperm cell making four actual sperms. So those are the general differences that you want to keep in mind, and similarities. So if you remember the Darren Natalie video, we saw, nice animations of Darren's sperm being developed, and the tube that first sperm went down is right in the middle here, this lumen of the tubules and the testes. And you can see all these up here are pre-sperm. And as they develop, they get closer to this tube. So we'll say this is developing. So make sure you're annotating this along with me. And since the sperm cells really don't have a whole lot of machinery to do simple things, they need helper cells. And that's what these Sertoli cells are for, to help sperm develop. Almost like a tutor or a teacher to the sperm, giving them what they need to be strong, to be healthy, so they can go out one day and be successful sperm. So similarities and differences. So different gametes, different amounts of cytoplasm, different number of gametes. So they both produce gametes, which are a fancy term for egg and sperm. They both use meiosis. And remember those hormones that started puberty in Darren and Natalie? You probably do. FSH and LH. So they both use those and those hormones. And oogenesis, I have an extra O there, so ignore that. Create cells with large amounts of cytoplasm, and spermatogenesis create cells with a little cytoplasm. So I can't see what was in the back there. They're different in that spermatogenesis continues throughout life, where in females, menopause, it stops. And spermatogenesis creates way more gametes. For a female, it's 400 to 500 eggs per lifetime. For males, it's probably billions of sperm throughout the lifetime. So spermatogenesis creates 4 gametes, where oogenesis creates 1. So 1 pre-sperm makes 4 eggs. For females, 1 pre-egg makes 1 final egg. And that's primarily because the egg needs so much cytoplasm. You got the half nucleus and you need a lot of cytoplasm to sustain that egg when it first gets developed. You end up getting these 3 little things called polar bodies along with that egg. And these polar bodies are basically a place to stick that extra DNA, and they don't ever get fertilized. If they did, it wouldn't matter because they don't have enough cytoplasm to sustain that new cell, with energy, mitochondria, all that machinery. So here we see the big size difference, okay, relative to each other. So we got the massive egg here, and you got this very small sperm. So you can see, yeah, you definitely notice a big difference, and all the more energy that this egg would have taken to be produced. So here we have a sperm trying to fertilize an egg. The egg is covered with this this layer called the zona pellucida. So we have, and it's very thick, it's hard to get through. So the sperm has something called the acrosomal enzymes that get released once the sperm hits the zona pellucida. Those enzymes get released. And it's just like the enzymes in any detergent you use. It's there to eat things up, proteins mostly. So the proteins are getting eaten and eaten and eaten, and once the sperm gets down to the membrane, the plasma membranes fuse. They fuse together. So what does that create? A nice little opening for the nucleus of the sperm to come in, join with the nucleus of the egg, and therefore go from 23 and 23 to 46. And you'll notice that the tail does not enter the sperm, so just the nucleus enters, not the tail. And then right after that happens, see these little, get to that in the next slide. These are little vesicles. This is for the cortical reaction. So once the membrane is breached and that nucleus is in there, these break open, and that's called the cortisome reaction, and that hardens. It hardens the membrane. See, is it listed here? It isn't. So it hardens, and that prevents any more sperm. So we look close up. This is the acrosomal enzymes here. We got the nucleus, and you have these little structures here. These provide power. These are the mitochondria. Okay, to help power that sperm journey. So as you create a little tree map similar to what I have here, what I just described, the acrosomal reaction. Enzymes digest the zona pellucida. Once the membrane is reached, the membrane is exposed, the tip of the sperm will fuse its membrane with the membrane of the egg, and the nucleus will enter. And the cortical reaction, vesicles in the membrane break open and then harden up, so no more sperm can get there. And that's really important. If more sperm got in there, you'd have way too many chromosomes, and it wouldn't be feasible. You really can't live without the exact number of chromosomes. Extras don't help. The exception is plants. Plants can have like double the number of chromosomes, and that's pretty fascinating, at least to me. Very cool seahorse. A lot of times people like to bring this up. They say the father is the mother. So you can see egg and sperm is exchanged, but instead of the eggs going to the female, the eggs stay in the male, and they develop in there, and then lo and behold, they hatch out of the male. Okay, so the male technically, not technical, it's almost like a little external pouch, kind of like marsupials, and those seahorses are born. So that's pretty cool. Here we see a blastocyst. and it's trying to implant into, I guess, where is this tissue? What is this tissue? What organ is this? It's the uterus. In our video, when we saw, what's her name? When they're doing the painting, rock climbing, rappelling, anyway, that video, we saw this happening, and that rock climbing was supposed to be a metaphor for the egg tumbling down the side of the woman's uterus until it gets a nice spot, and it is immediately going to want to burrow into this tissue to find blood. Why does it want to find blood? Because blood will give it nutrients, right? Nice rich blood. Get sugars, proteins, water, oxygen, and be able to release its waste products. And here's this nice little kind of showing you the development of an egg after it's fertilized. You have it splitting in half, and then we have another stage, and this will be the blastocyst where it implants into the uterus.